that once you set your foot in life sciences, you start your love and hate relationship with this little creature, mouse. So this is the beauty of it, because the gene therapy is becoming so personalized. They want to treat every single mutation.
来自于赛业苏州生物科技有限公司的董事长，还有清华珠三角研究院人工智能创新中心主任的这个韩兰青教授，他给我们带来的题目是 HUGO-GT 全基因组人源化模型加速罕见病基因治疗的研究啊，请。
Yeah. Okay. So this is the last talk for the day. I guess the energy level is getting lower, but I hope you guys stay on because this is going to be very relevant to you guys. So today I'm going to talk about the mouse models. Why mouse models? Well, actually, Professor Li and Professor Gao, we were in the same meeting last month in Tsinghua University in the medical school. So we were sitting together over lunch, and the dean of the medical school was telling me, hey, you know, I'm recruiting young talents from overseas, and I'm having such a headache. And I asked, what headache? And they're negotiating with me. And I asked what are they negotiating with you for? Salaries? No, he said, never salaries. They negotiate about the number of cages they're going to get when they are here. So see, this is how important this little creature are to you guys. I say it to the young people sitting in the back, that once you set your foot in life sciences, you start your love and hate relationship with this little creature, mouse. You love it because you cannot do any research without it in life sciences, in drug discovery. But on the other hand, you hate it because more often than not, they cheat on you. They mislead you into believing something that is not true. So this is when Cyagen comes in and tries to fix at least some of the problems. Okay, let me. So this time, we're bringing in the HUGO-GT Humanized Genomic Ortholog mouse models to accelerate gene therapy. So at least we're going to fix some of the problems, especially in gene therapy. Because in life sciences, especially in drug discovery, what you want to achieve is to find the right drug for human, not for the mice. So you want to humanize the mice in many different ways and to different levels. So how to do it? I would say as a clinical researcher, sometimes you see a mutation running in a family and you think it's very interesting to study, and you just simply put that mutation into the mouse. Does it work? From my 15 years of Cyagen, I'm telling you, most of the time it doesn't work. Because, yeah, the two species share 99%, 97%, sometimes say 97% of the gene, but that, the truth is that 97% of the gene can be traced to the single ancestor. But if you look at them base by base, they're different. So sometimes you put the disease-causing mutation in human into mice, it don't work for various reasons. And what do we do? So I'm going to switch to the next slide. So I was talking about the, TG, point mutation. Okay, the point mutation is the simplest humanization. You put in the disease-causing mutation in human into the mice. Okay, and then what? TG, transgenic, right? And well, there is a CDS humanization. Basically, you replace the mouse gene with the human gene, but only the CDS gene, only the protein coding sequence. That works pretty well when you're developing small molecule drugs or protein drugs. Well, let's just give you an example. For example, if you're developing a PD-1 drug, you only need to humanize the PD-1 gene on the protein level. So you can just put the human PD-1 coding sequence into the mouse. At the same time, knock out the mouse PD-1. And that's it, because, well, you're either developing a small molecule drug or you're developing, most likely, the antibody drug. You're targeting the protein. But in the area of gene therapy, now we're talking about the rare genetic diseases, right? In gene therapy, most likely, you are targeting the nucleic acid, the DNA or the RNA. In this case, you only humanize the protein. It's not sufficient. So that's why we need a better model. So if you ask the gene therapy people, what do you know? What do you use? For example, for people who develop the SMA drug, this, Zolgensma. Well, Zolgensma is different. I mean, Spinraza, okay, for the ASO drug. What model do they use? Most likely, they are using the back transgene. But I've heard a lot of complaining about the back transgene model. I hope there's no people in this room. Anyway, because of what? Because the back transgene is integrated into the mouse genome randomly. You have no idea where it ends up with. And sometimes with multiple copies, right? And what you see, you think is a phenotype. It might be caused by the disruption of the genome at the insertion site. You have no idea. That's why I heard a lot about, a lot of the complaints about this transgene models you can get access to from, So this is why we came up with a new approach. We think it's a revolutionary approach a few years back. We call it HUGO-GT. HUGO stands for Human Genomic Ortholog. So this approach has two steps. First of all, you humanize the gene on the genomic level. Okay, so let's say you want to humanize the gene. You replace the mouse gene with the human counterparts. Not just the CDS, but the whole genomic sequence with the exon, intron, both ends of the UTR intact. And sometimes you might even want to humanize the promoter, if you're targeting the promoter. Okay, so, well, I'm not going to get into details, but what I was talking about was you need a better model for gene therapy because you're targeting DNA or RNA. That's the first step. You humanize the gene, and what we do is that, I say our chief scientist, Dr. Ouyang, is here, so he's the expert in this technology. He developed this technology, a few years back. So first of all, we humanize it, and we use the ES targeting. We didn't use CRISPR at all. I'm going to tell you later why we cannot use CRISPR. It's an ES targeting technology, okay? And after the first step of humanization is done, you have the ES cell with the gene humanized on the genomic level, and you call it wild-type humanization. Why wild-type humanization? Because there's no disease-causing mutation as yet. And once you have that ES cell with the human gene in place, then what you can do is you can easily introduce whatever disease-causing mutations you want to put into. So that's the beauty of it. Okay, so you can see that we put some focus on neuromuscular disease, ophthalmology, and metabolism. So these are the genes we have fully humanized. So about 2 years ago, we selected about 100 genes that are frequently studied in rare disease gene therapy. So let's go over them one by one. For neuromuscular diseases, you can see that we have humanized a whole bunch of genes for Alzheimer's disease, Parkinson, Dravet, ALS, and more. I'm not going to list them one by one. For motor neuron diseases, we humanized the DMD gene and the SMN for SMA, right? So most likely, because we're humanizing on the genomic level, it's much bigger. So it could go up to a few hundred K easily. For example, a few weeks back, we initiated a new project for this company who asked us to humanize the whole US H2A gene, which is for the Usher syndrome, right? For the eye disease. And that gene alone is over 800K. So think about it, humanize the gene with 800K. And that's the first step. And then we're going to introduce a whole bunch of disease-causing mutations in that gene. With CRISPR, can you do that? I was told by a lot of people that they could go maybe as high as 20K at a time. If it is over 20K, they couldn't even, make it. So in our case, we can easily go up to a few hundred K. So if you look at this, the list, we have the APP gene humanized, we have the tau gene, the MAPT, PSEM1, and a whole bunch of them. Yeah, and we're not just making the mouse models. At the same time, we also put the mutations into the human iPSC cells. But this is not the focus for today. Okay, SMA. SMA is different. SMA, the human has two copies, right? So one is the SMA1, the other one is SMA2. And, we function because we have SMA1. So SMA2 is kind of, you can consider it as a defective copy because it's got a mutation. So one way to treat SMA using ASO is to correct the splicing of SMN2 to make it like SMN1 so that it could produce enough the right protein, right? So this model, what we did was very straightforward. We just replaced the mouse SMN. There's only one SMN gene in mice. We replaced this gene with human SM2. And that's it. Very straightforward. So people they couldn't believe it because you couldn't, understand why that the back, the transition is so complicated, And in our case, it's very straightforward. And we tested with our homemade Spinraza. Well, Spinraza is too expensive if you buy it off the market. I was in Ionis last year giving a lunch and learn. So 60 people came in, and I gave the talk about our SMA model. And they all agreed that this is much better than that they used to develop the Spinraza. Okay. So you can see that, there are a lot of advantages, but I'm not going to get into details. You can see with the treatment, so the mouse just lives forever. Well, actually, I'm only showing here the Spinraza, but actually we tried all three drugs: Spinraza, Zolgensma, and risdiplam. They all work pretty beautifully. So here you can see that the phenotypical differences between the heterozygous and homozygous, and also the wild type. And so you can see very prominent, significant phenotypical changes once the mutation is there, once the human gene is there. And then this is, okay, we're looking at the brain, the spine, and this is the ASO treatment by ICV injection. Yeah. And also the tau. Well, I'm only showing the the ASO treatment because we're still gathering data from the other two treatment, but the results are very promising. Actually, we've did drug efficacy studies for a few of our clients who are using the AAV-based treatment. Okay, so I'm going to go over quickly some of the HUGO models. Some of them are not fully validated yet, but they're on the way. So this is the tau gene, and you can see that the humanized mice doesn't carry the mouse tau gene, but the humanized one has the human tau gene, but no mouse tau gene. Okay, so this is very clearly shown here. And this is TARDBP, the ALS gene. And you can see that, this one is actually this one, yeah, this one is the tau, the humanized gene with mutation. So you can see plugs here, the aggregation of the toxic protein here. And MECP2. Well, this is implicated in Rett syndrome. So we humanized the MECP2, and also we put in the most prevalent mutation in there, and we did a test. Yeah, so the mutation is T158M. So you can see there's some behavioral differences, but we're still going to do a lot of other tests. So this is still ongoing. But somehow the Rett Syndrome Foundation, they found out about it, so they contacted Cyagen. So we shipped a few mice to them. Now they're asking us to put in a whole bunch of mutations one by one. So this is the beauty of it, because the gene therapy is becoming so personalized. They want to treat every single mutation. Like, early this year, the Cystic Fibrosis Foundation contacted us because they knew we had the CFTR humanized. And cystic fibrosis, 80% of the patients have the Delta 508 mutation, if I'm not mistaken. But they also have a whole bunch of other genes, many of them, that account for the rest 20%, right? So the foundation is asking us to put in a lot of mutations into CFTR. You know why? Because I saw on their website, they just gave a $15 million US grant to Prime Medicine, the company founded by David Liu. So I'm pretty sure they want to use our mice to use Prime, to be treated with the Prime editing technology. Yeah. So, I'm a little bit off the topic. And here we're showing a few additional genes. This one for Parkinson's disease, this one for spinal muscular atrophy with respiratory distress type 1. This one is spinal cerebellar ataxia. And for ophthalmology. Okay, so ophthalmology, I'm going to give an example on the rhodopsin gene. The rhodopsin gene, we have it fully humanized, and we put in this famous P23H in there. And you can see, here, a pretty big difference of the A wave and the B wave. For people who study ophthalmology, you know it. I have no idea, but if you're interested, we have our scientists in ophthalmology sitting here. Okay. And the ASO treatment. So this drug has not been approved yet. So we took it from the patent, we got it synthesized, and we applied it into the mice. And you can see this is without treatment. You can see the, big phenotypical differences. But with the treatment, you can see big improvement. So I believe this model, we've sold quite many of them, to the gene therapy companies, I guess mostly in the US. This one, ABCA4 and USH2A for the Usher's syndrome. So this one, I'm telling you, this one is 800K. So to humanize the 800K, you can ask anybody else to try it. It's a big task, I'm telling you. But this is not our limit. I actually, not too long ago, I challenged my team to humanize the full DMD gene, the dystrophin gene of 2.2 meg. I don't know if I can do that. And I just want to use it as a showcase that we can humanize the biggest gene in humans, so we can do whatever. And, okay, metabolism. So this is the TTR. And this is siRNA treatment. This is with one of our customers' consent. We're showing here the early drug efficacy studies we've done for one of our customers. So you can see this actually quite effective. Yeah. Oh, this one. This one is, I don't even know how to pronounce it, but this is one of the rare skin diseases. Let's just call it EB, okay? So this is caused by a mutation on the type 7 collagen. Yeah. And you can see very severe, phenotype. And this one, I think we are also shipping it to some of our customers, to do the drug efficacy studies. Oh, here, I almost forgot. So we have this program. We started with selecting 100 genes and make them in-house. But, we have how many? 7,000, 8,000 rare diseases? And I believe we have the same number of genes, at least, if not more. So ideally, this is our vision. We want to have an ideal model for every single of them. But we only made the first 100. So I want to, have the researchers, drug development companies join us to develop more of this humanized mice. Because in gene therapy, it's going to be more and more personalized. You might just want to target that particular gene, that particular mutation. That's why, this approach, you can put in whatever mutations in there very quickly, very efficiently. And as we speak, this new type of models are gaining traction. Okay, so lastly, we are bringing to this event Rare Disease Data Center. This is a database we developed in collaboration with Tsinghua University. So, a few years ago, actually, I myself I don't have a biology background. And also, I'm not a bioinformatician. But sometimes I want to find out things. So I go to NCBI website, I go to Ensemble, but it's very difficult to use. So I want to develop something that is very easy to use, very user-friendly. And you come to this one single place, you have everything. So basically, you can find everything about rare diseases, genes, mutations, animal models, clinical trials, drugs under development, everything. So I encourage you guys to go on Rare Disease Data Center. Actually, you just put this forward into Baidu. It's going to lead you to our website. Or, you can just type in RDDC, which is short for Rare Disease Data Center. And it's going to, Baidu is going to find it for you. And I encourage you to go and play around with it and let us know what you like and what you don't like. So this is a not-for-profit. So this is something that we want to bring to the community for free, forever. So, this is, I'm not going to get into details. My time is running out. Here I showed some of the functions. So basically, what is good about this database is that we have quite a few AI models. So if you have some mutations that are marked as VUS, you can put it in here, and it's going to give you a score after running the AI model, how likely it's disease causing. And another model is going to tell you if it is going to cause the splicing problem. Because from our experience, about 10 to 15% of the mutations are actually causing the splicing problem. But I heard from another, presenter this morning that the percentage could be much higher. Well, it's not shown here, but the other feature we just added is we put the large language model into it. If you don't know how to use it, it doesn't matter. You just talk to it like you talk to ChatGPT. And so my vision is that not too long from today, researchers, clinicians, bioinformaticians, and the patient, or parents of patients can easily go on and use. Yeah, so you're welcome to use it and give your comments. Okay, thank you very much.
好，谢谢这个韩董事长跟我们分享了这个人源化的这个全基因组的基因 therapy。所以我刚刚注意你的 logo 上面 GTY 是什么意思？就是基因 therapy 的意思。
基因 therapy， 对
对。那有什么问题？
我没注意到我们那个唯一的老外都走了，实际上我应该说中文。
那个韩总，那个非常精彩的这个介绍。然后呢，我是药明康德，然后今天还有我们2位同事也一起来了。然后我可能会觉得说，在业的这个工作是非常有意义的，至少从某一个层面上来说，就是您刚刚提到的对于这种一些非常经典的TG model 的这样的一个，我个人觉得可能有一些垄断的这样一个场景做的一些比较有意思的工作。但是有一个问题，就是我们在跟我们的客户在交流过程当中，他们可能会对于这些我们新开发的这些 humanized model 的接受程度呢，可能跟不太一样。他们可能还是觉得那些发表过 paper 或者说验证过的这种东西会更有
对
这个接受度。
这是一个你提的问题特别好，就是说比如说我们的 SMA model，
对，
因为前面已经有 approved drugs， 所以说可能别人要是用，就是已经有这个 track record 这些模型的话，就可能他觉得更安全。我们确实是碰到这种，就是说，但是有的人呢，他会希望能够用一些新的东西啊，因为就是说这个东西为什么好呢？实际上我们就是做了这么长时间以后，我们是自己发现啊，它实际上呢，它有很多的好处，我们是以前没有预见到的。你想想看，你假如说你把它插在一个不知道 genome 上面，不知道什么地方，它是没有上下文的。你一个基因它发挥作用，实际上它在不同的细胞、不同的这个 cell type 里面，它的表达是不一样的。它是会有 alternative splicing， 它的 expression level 也是不一样的。你硬把它塞在一个 genome 的任何一个地方，它是不会给你一个正确的一个表象的。你看到的可能是一部分是你想要的，还有一部分呢就是 artifact。但是我们这个呢，它是还是就是说，当然人和老鼠不一样啊，就是说它是已经是最大程度了接近了人。我觉得可以这么讲。所以说对于这个开发药物，对于它的这个成功率啊各方面都会有好处。所以说呢，目前我们这个主要的这个业务还是在美国啊，因为美国做这个就是创新的 gene therapy 还是多一些。你像这些大公司就开始用我们了，像 Sarepta、Ionis、这个 BioMarin 这些 you name it 都是我们的客户。
是吗？
对。
对。那可能就第二个问题，就是您刚刚提到有一些很多CNS 相关的这种 humanized model，
对，
它会有一些特殊的地方，比如说它可能会在这个人源化的这个就是人的基因前面会 引入一些比如说属源的这个神经系统特性的这个 promoter 在前面，这样可能会对于它的表情出现会更加容易或者更加严重一些。就拿那个 TDP43来说，但是如果是用我们现在这个 HUGO 的这种方法，它可能会要分2步走，然后或者说这个时间线会很长，不像 TG 一样，就是一把就可以搞定，只是它在方的筛选上会可能时间比较久一些。
对，但是呢，你要注意一点啊，就是为什么有很多的这个 TG 或者 back transition， 那是因为可能100个里面99个失败了，有1个成功了，结果呢就被存在了里面。所以说，实际上这也是一个误区。我们以前也去做这个 transition TG， 但是一做就不行，一做就不行，然后你筛选的这个过程也非常的繁杂。所以说你要是自己去碰的话，那个机会就是成功的几率是不大的。
OK。
最后呢，就是说只有成功的人，就好像说赌钱一样，你怎么觉得这赌钱的人都赢钱啊？因为他输钱的人他不告诉你。对，所以有这样一个问题。所以说呢，这个你不要以为 TG 就是很容易 ，TG 实际上那个工作量，你去把它筛选出来，有好的表情，那个工作量是不低的。
是的。OK。谢谢韩总。
好，谢谢。
好，最后一个问题，时间问题。
我可以刚才补充一下这位尤老师提出的问题，就是说，我认为这个人源化的模型跟这种常规的模型有一个非常非常大的优势。比如说，你要做的是 ASO， 做的是 siRNA， 做的是 CRISPR 的盖的 RNA 等等，你必须得把像这个人的序列是最好的。当然，你可以说我临床上这个人的序列在小上用一个替代物，这种也可以，但是毕竟对吧，不能反映很多真实情况。你的真正的效率怎么样？体内你是没办法用这个替代物来去模拟的。但是如果我们把这个鼠的序列人源化之后，我先不说表情如何，单从这个基因的一个编辑效率、敲剪效率等等，我就可以拿到一个体内的真实结果。这种如果你不做人源化的话，你基本上是不可能拿得到的，对吧？这是至少以上一个优点。再一个优点是，你这一个模型如果它既能够反映这一个基因层面的一个敲剪编辑效率，又能反映它对于生理功能的一个改善，这种也会比你传统的方式在什么类器官啊、细胞上啊，只看到一个某种生化层面的效果要好得多。我觉得这个是人源化的模型所无法取代的。
这是我们那个赛叶的负责基因治疗的任博士，那个因为他是做了很多给我们的客户的这个药效的项目，所以说他在这方面还是很有体会。
好的，今天很好啊，我们围绕着这个罕见病不仅体外实验、体内实验，我们就是其实就刚刚围绕的话题就围绕着我们向体内最可能的大的模拟，我们从细胞前面有细胞模型，还有动物，当然小动物、大动物，我们对，然后这个重要就是罕见病的基因治疗方面得到很好的总结。好，今天我们下午的讨论会到此结束，谢谢大家，谢谢讲者，也谢谢听众，谢谢。